In this second video for lecture 4, I'd like you guys to really start to think about the eukaryotic cell as a whole. And we're going to start to think about this eukaryotic cell as a processing center. Now we're going to go through each one of these individual organelles, and we're going to talk about their individual functions. But it's possible for you, if you understand how the cell works, to connect the function of any particular organelle to the functions of another. I'm going to ask you to do this not only on the first exam, but I'm also going to ask you to do this throughout the semester and on your cumulative final exam, because that's a fantastic way for you to tell me that you understand how the cell works as a whole. We're going to start by taking a look at the endomembrane system. Endo means internal. And that endomembrane system is a series of membranes, both connected directly and indirectly, that performs a really critical function for the cell. Now, this endomembrane system begins with the nuclear envelope. An envelope is a double bilayer, which we're going to talk about in just a moment. And that nuclear envelope is what surrounds and encases the nucleic acid genome, the DNA genome of the cell. And so this endomembrane system is going to be found only in eukaryotic cells. The endoplasmic reticulum is next. This is the site of protein synthesis for most proteins. And this is going to be directly connected to the nuclear envelope. There's a Golgi apparatus which sorts and packages those products synthesized by the endoplasmic reticulum. And this is indirectly connected to the plasma membrane. I've crossed off plasma membrane here because even though it's directly connected to this endomembrane system, the plasma membrane is the outer layer. So it's not truly an endomembrane, but I don't want you to completely disconnect it from this entire process. Now, all of these components are going to be connected, again, either directly, for example, the nuclear envelope is continuous with the ER, or indirectly, via vesicles, which are little membrane-bound sacs, little round spheres surrounded by membranes that can actually disconnect from one organelle, carry their contents to a new one, and then merge their membrane with that new organelle. These vesicles, these little membrane-bound sacs, are a critical component for trafficking components through the endomembrane system. Now, the nucleus, again, this is found only in eukaryotic cells, but this nucleus is not only the core of the cell, but it's also going to be the heart of this endomembrane system. It's the area that actually contains the DNA nucleus in a eukaryotic cell. Now, there is going to be some other nucleic acids. This isn't the only location of nucleic acids in the cell. Not only are we going to be creating RNA copies, but there is actually going to be a small nucleus or a small amount of nuclear DNA found in some endosymbiotic organelles. So if you've taken Bio 1 at this university or another biodiversity type class, you've probably learned about the fact that mitochondria and chloroplast actually used to be individual cells. Since they used to be free living cells, they have a nucleus or a nucleic acid genome of their own. I'll show you an example of that in a minute. Now, this nucleus is going to be surrounded by a membrane, which is actually a double membrane. So since each membrane is composed of a bilayer, bi meaning two, that double bilayer is actually going to have 4 layers of these phospholipids. Embedded inside of this phospholipid bilayer is going to be nuclear pores. So again, membranes are not just dividers, they give functions. Nuclear pores are going to be protein complexes that allow material components from the nucleus to get out or to get from the cytoplasm into the nucleus. But the proteins of these pores are going to carefully regulate what comes out and what goes in. What you can see here is this wrapping type effect. This is actually why this nuclear envelope is a double bilayer. Each one of these lines represents a bilayer, And so where these fold around near the pores is what actually creates the double bilayer. Underneath of that nuclear envelope is another layer of protein called a nuclear lamina. And this nuclear lamina are built of protein filaments that maintain that really critical shape of the nucleus. And it's why anytime you're looking at a nucleus through microscopic images, it's going to look like a really pretty perfect circle. So here is our nucleus. It's formed of a double bilayer where you see this wrapping of this envelope. It's continuous. This bilayer of phospholipids is continuous with the endoplasmic reticulum, specifically the rough endoplasmic reticulum. Inside of this double bilayer is going to be embedded proteins called nuclear pores. There are also going to be filaments of protein that are underneath of this nuclear envelope. These nuclear lamina is going to be a component that maintains the shape of the nucleus. And then inside, internal, is going to be the nucleic acid genome of this cell. Now, that DNA genome is going to be composed in one or more pieces called chromosomes. You've probably heard this term before. Humans have 46 chromosomes, 23 different pairs, because we have 46 pieces of DNA. Each one of those pieces is a single DNA molecule, a single DNA polymer, and it's associated with proteins called histones. So there's another function of these proteins to compact this DNA down into a shape, an organization that can actually fit inside of the nucleus. Now, surrounding this nucleus is going to be an endoplasmic reticulum. The endoplasmic reticulum is folded. So unlike the nucleus, which is surrounding a nuclear lamina holding this shape, the endoplasmic reticulum is specifically folded because this creates that large surface area, which is so important in eukaryotic cells. We call these folded, flattened shapes cisternae. And these cisternae create that large surface area that gives the endoplasmic reticulum its function. We're going to talk about two different sides of this endoplasmic reticulum. The rough endoplasmic reticulum, which is the largest of that endoplasmic reticulum in most cells. It's rough because, as you can see here, it's actually dotted by these little tiny dots. It turns out these are ribosomes that are actually bound to the surface of the endoplasmic reticulum. The smooth endoplasmic reticulum does not have ribosomes, and so the smooth endoplasmic reticulum will not synthesize any proteins. Now, this rough endoplasmic reticulum accounts for about half of all of the membranes in the cell. When we talk about this surface area, it's very, very large. And again, it's going to be continuous with that nuclear envelope. We call the inside of the ER a lumen. And it's important that we name this space because the cytoplasm, which is that outside filling inside of the cell, is actually never going to mix with the inside of the endoplasmic reticulum. And so the lumen and the cytoplasm are two completely separate spaces. So that membrane, in addition to having its own functions, in addition to having that surface area, is a wall that divides the lumen of the ER from the cytoplasm. Now, that rough ER is going to have ribosomes that attach to the surface of the ER. And so one function of the endoplasmic reticulum, the rough endoplasmic reticulum, is to synthesize those proteins. The proteins that are made on the surface of the ER actually get placed into the lumen of the ER. And from that inside of the lumen, they can have carbohydrates that are attached to them. Remember that the bond that forms between carbohydrates, between monosaccharides, is known as a glycosidic linkage. So we call proteins with sugar groups attached to them glycoproteins. The only place which these glycoproteins are produced is in the lumen of the ER. So the only ribosomes that make glycoproteins are going to be rough ribosome or rough ER ribosomes, so ribosomes attached onto the endoplasmic reticulum. these will also, as a function of this rough ER, create vesicles that distribute and transport these materials to the Golgi. The smooth ER has no ribosomes, so there's no production of proteins, but we do synthesize lipids, including phospholipids and steroids. We also detoxify drugs and poisons, and we can store calcium ions, which you're going to see again when we talk about how cells communicate. When we think about the function of these different organelles, you can also connect these functions to cell types. For example, one of the jobs of your liver cells is to detoxify proteins from your body. And so I hope it won't surprise you to learn that liver cells have large amounts of smooth ER, because the smooth ER in the liver cells is what actually does that detoxifying. But there are also going to be cells that are responsible for the production of hormones. And since some hormones are steroids, well, if you have a cell that's responsible for the production of these steroids, that would also be an example of a cell that has lots of smooth ER. Now, I want to introduce you to ribosomes, and even though we're going to talk about ribosomes more as we get into the genetics component of this class, these ribosomes are critical for understanding the functions of that ER. Ribosomes are actually composed of two different macromolecules. They are enzymes, so they're made out of proteins, but they also have a component of RNA. So these are complex structures responsible for the synthesis of peptide bonds in the formation of proteins. They can be found in two locations inside of the cell. The first we've already talked about. These are bound ribosomes attached to rough ER. But there are also free ribosomes that float around in the cytoplasm. They still make proteins, but because these proteins are being produced in the cytoplasm and not in the lumen of the ER, they will not be glycoproteins. Here's an example in another picture for you of these ribosomes, including free ribosomes floating around in the cytoplasm, producing traditional proteins, and bound ribosomes attached to the rough ER, synthesizing glycoproteins. Once material are made in that ER, whether they're produced by the smooth ER, such as lipids, or if they're produced in the rough ER, such as glycoproteins, they can be packaged into vesicles. Vesicles can have proteins on the inside, membranes on the outside, so both components of the smooth and the rough ER can be packaged into these vesicles where they move to the Golgi. Materials are received on the cis phase. Cis actually means facing. So this is the face of the side of the Golgi that faces the endoplasmic reticulum, and it receives the incoming material. It then packages that material through the layers of the Golgi, modifying and sorting those materials as they go. And when they leave the transface of the Golgi, they can be placed into new vesicles with new component arrangements. And this allows us to start to create some specialized vesicles, which we're going to be talking about in the near future. So here you can see these glycoproteins as they're moving through this secretory pathway. In the center of the cell, we have this Golgi structure. In the center of the cell, we have this nuclear structure here. And as the cell moves, you can really kind of see that boundary. Again, this is a really beautiful circular shape because we have a nuclear lamina, that protein structure, which is supporting the cell. And we also have this Golgi structure. Now, the ER, as you'll recall, is a very large network of many, many membranes. It's about half the membranes in the cell. So you're going to see that it seems as if these proteins are trafficking from many locations in the cell, but this is just that expansive network of the endoplasmic reticulum. And they're going to be moving to that centralized location, which is the Golgi. So the nucleus contains the information of the DNA genome, and then allows that storage of information to eventually be translated into the information of proteins, which are created by the rough ER, We have membranes by the smooth ER, which are placed into vesicles together. And then these are trafficked to the Golgi, arriving via the cis face and leaving via the trans. So here's an example of the Golgi. Would this be a light or an electron micrograph? Yeah, good. This is taken by electron microscopy. And would you say this is an example of scanning electron microscopy or transmission electron microscopy? Good. Hopefully you said this is an example of transmission electron microscopy, because we're not seeing a three-dimensional shape like this. We're actually seeing details as if we took a slice through that Golgi. Now, this is going to be indirectly connected to the rest of this endomembrane system, not directly connected, which is why you can identify this little unique shape here. So now I'd like you to answer this question, because the order of this path of the endomembrane system is very important. Which one of these would describe the path taken by a glycoprotein that's destined for the plasma membrane of the cell? Good. Hopefully you were able to recognize that membranes are going to be synthesized in the smooth ER. And so we don't need C or D here because we're not dealing with a membrane. We're dealing with a protein made in the rough ER. These are going to go to the plasma membrane, which both of these options allow for. And so the decision becomes, do we arrive at the cis to the trans, or from the trans to the cis face of the Golgi? Cis means facing, or you can remember that C comes before T alphabetically, and so we go from the rough ER to the cis Golgi to the trans Golgi to the plasma membrane. Now, the endomembrane system as a whole is going to be a multi-component part of the cell. It goes from the nucleus, that storage central component for all of the genome, through the ER, where we're actually producing necessary components of the cell, moving it into the Golgi, where we can further modify those components and place them into vesicles where they can be shipped to other sites. I do want to have a couple of things that I emphasize to you. The first is that the ER lumen and the cytoplasm never mix. So anything that's produced into the lumen of this ER is actually going to stay luminal. So something that's made in the lumen of the ER, so something that's made inside of this ER, say here, is going to become an interior component of this vesicle. And then when this vesicle actually merges with the Golgi, it stays luminal, it stays interior. So something that's cytoplasmic stays cytoplasmic, and something that's internal stays internal. When this internal component of one of these vesicles merges with the plasma membrane, now those components get released. So cytoplasmic stays cytoplasmic, but interluminal can actually become extracellular. Now, ribosomes are also going to be a component of this endomembrane system, but those ribosomes do not get trafficked. So yes, there are ribosomes, we can see them here studding the surface of the rough ER, but those ribosomes are only going to make their proteins and then go back to doing what they're doing. Ribosomes have no interaction with vesicles, no interaction with the Golgi. And so we're not actually trafficking the ribosomes themselves. What we're trafficking through the endomembrane system are the products produced. There are a large number of different proteins and components that can be made. And so when we take those specialized components and place them together, we can actually recognize the fact that vesicles can become specialized. When these vesicles get specialized properties, we call them vacuoles. And there are a number of different vacuoles that can be formed that have specialized properties, such as food vacuoles, contractile vacuoles, or a central vacuole. Here's an example of a contractile vacuole where the cell is actually expanding this vacuolar space, which allows it to be able to pump water out of the cell. So it's contracting and pushing the water out to regulate its osmotic pressure. Central vacuoles are found in plant cells only. They're a large central component that stores water. This is the reason why when you water your cell, it looks all plump and tight and beautiful. But when you don't water it, it gets all saggy and sad. Some vesicles that leave the Golgi can become lysosomes. These lysosomes are going to be a specialized vesicle that contain hydrolytic enzymes responsible for digesting macromolecules. So go ahead and tell me in Socrative, hydrolytic enzymes break down macromolecules via what process? Well, if you break these words down, you can start to have a better understanding of their purpose. Specifically, A, dehydration synthesis. Well, synthesis means to create new, right? To synthesize something. So synthesis is not going to be a component that we use. Even hydrolytic synthesis, which sounds like hydrolytic enzymes, would describe new bonds. So that's not it. So we have hydrolysis and dehydrolysis. But hopefully you remember from last class that hydrolysis is the breaking down of bonds, which involves the introduction of a water molecule to separate them. So dehydrolysis would be the removal of water. Dehydration synthesis is when we make a bond between monomers, and hydrolysis is when we break that bond. Hydrolytic enzymes are what kind of macromolecule? Well, since enzymes are proteins, hydrolytic enzymes will also be proteins. Now, lysosomes are going to be another example of how we can get some specialized vacuoles. When we actually do phagocytosis, and I'll show you a little video shortly, phagocytosis is the process by which the cell engulfs food in its environment. And this creates a very large vacuole known as a food vacuole, and it's going to contain material from that cell environment that's been engulfed. But when we take food and we need to break down food, just like food in your stomach, we're going to use acid, like your stomach acid, and we're going to use enzymes. Well, hydrolytic enzymes from lysosomes are perfect for this. And so we can actually merge that food vacuole with a lysozyme that contains those hydrolytic enzymes and acid to be able to break down and digest the food that was just engulfed. So here's an example of a cell that's going to be phagocytosing a bacteria. Immune cells use phagocytosis as part of the process by which you can protect yourself. So here's your immune cell. The bacteria are labeled in green, and there's the engulfing of that food particle. And as you watch that green disappear, that's actually because hydrolytic enzymes inside from the lysosomes of that cell have broken down that food particle, which in this case was a bacterial cell.